Hello students, today I will discuss about the auxin and its role in growth. First of all, we have to know that what is growth and how a seed can develop into a tiny plant or a big tree. Growth, irreversible change in mass is known as the growth. During growth, new cells are added by the cell division. Continuous cell division in the meristematic regions takes place, and after this, the enlargement of these newly formed cells takes place. Due to the enlargement, the cell size increase. After this, the differentiation of these cells takes place into different types of tissues, and these tissues together form organs. During this, synthesis of new cellular material and organization of subcellular organelles also takes place. तो जो इसका परिणाम होता है the ultimate result is increase in drive rate. And what is the development? Irreversible change in state. As we know that plant continue to grow throughout their life cycle, their lives. Different type of stages comes in the life cycle of a plant, just like embryogenesis, juvenile stage, adult vegetative stage, and adult reproductive state. So we can say whole series of changes is known as the development. Basically, development is associated with morphogenesis and differentiation. Hormones. It is well understood that growth is regulated by the specific type of chemical substance which are formed in the plants naturally. Word hormone comes from a Greek word harmeo, which means to stimulate. According to Pincus and Thyman, organic substances produced naturally in the higher plants controlling growth and other physiological functions at a site remote from its place of production and active in minute amount. Starling first time used the term hormone, and these hormones also known as the phytohormones. Phytohormones named because एनिमल्स ऑल्सो प्रोड्यूस हॉर्मोन्स. तो एनिमल जो हार्मोन होते हैं उनसे उनको अलग डिफ्रेंशिएट करने के लिए फाइटो हॉर्मोन्स भी कहा जाता है. दे आर ऑल्सो नोन एज द ग्रोथ प्रमोटर्स एंड ग्रोथ रेगुलेटर्स. बट growth regulators are those chemical substances which are manufactured in the laboratory by chemical method. Plant hormones can be categorized into two groups, growth promoters and growth inhibitors. Both are play very important role in the plant growth. Growth promoters are auxins, cytokinins, gibberellins, and growth inhibitors are ethylene and abscisic acid. Characters. When we talk about the different characters of these hormones, we can say that they are usually produced at the tip of root and shoot or at the meristematic regions, and the transfer of hormones from the producing part to other part takes place with the help of phloem, or we can say that phloem is responsible for the transportation of these hormones. Their growth promoting action occur only when they are used in definite quantity and concentration, and they are required in traces. यानी कि जो हॉर्मोन्स होते हैं ये बहुत ही कम मात्रा में ही इस्तेमाल किए जाते हैं. They can be isolated from the plant organ by the chemical method. Discovery. When we talk about the discovery of auxin, it was first suggested by Julius von Sachs in the later half of the 19th century. Although Charles Darwin was the first who acknowledged the existence of plant growth hormone, while working on tropism in plants with his son. He demonstrated the bending of coleoptile tip of Canary grass towards the unilateral source of light. And what is coleoptile? In monocots, the plumule in the seed remain covered by a protective sheath which is known as the coleoptile, just like rice, maize, avena, etc. When a unilateral source of light is kept, there is a bending of coleoptile tip towards this source of light. If the tip of coleoptile is cut off or can be covered by aluminum foil or tin foil, what will happen? There is no bending at all. So by these experiments, he concluded that when seedlings are freely exposed to a lateral light, some influence is transmitted from the upper part of the plant to the lower part, causing the latter to bend. इसका मतलब कुछ एक ऐसा सब्सटेंस होता है अपर पार्ट में जो लोअर पार्ट में या दूसरी शेडेड साइड में ट्रांसफर हो जाता है और वो रिस्पॉन्सिबल होता है कर्वेचर के लिए. After many years, Boyson and Janssen conduct a experiment in which he capitated coleoptile from the tip and put a block of agar inserted between the tip and cut stem. Result was the curvature of the coleoptile towards the unilateral source of the light. In picture we can see that here the tip is cut off and a gelatin का जो पीस होता है वो उसके बीच में रख दिया गया और रिजल्ट क्या था? जब unilateral source of light दिया गया तो bending और curvature of the coleoptile takes place. In another experiment, what he did, उन्होंने क्या किया कि माइका एक इसको ट्रांसफर किया और कोलेप्टाइल टिप में और एक माइका का जो पीस है उसको वहां पर प्लेस कर दिया और जब यूनिलेटरल सोर्स ऑफ लाइट दिया गया तब भी वहां पर कर्वेचर हुआ. In the another experiment, Paul cut off the coleoptile tip and he placed it asymmetrically on the cutting head of the coleoptile and he discovered that coleoptile bent away from the side with tip and he concluded that tip secretes a substance which promotes the growth even in the dark. When was successfully isolate the growth substance from the avena coleoptile tip. He cut the avena coleoptile tip and placed them on the small agar block for small time. After that, he placed these agar block on the tip of the coleoptile and he placed them asymmetrically. There is a bending of coleoptile tip. He placed agar blocks asymmetrically on the decapitated coleoptiles. Typical curvature was there even in the dark. And this bioassay is known as the avena curvature test. Auxin is first naturally occurring phytohormone. Kogel and Smith found that human urine is rich in growth substance and they concentrated human urine by series of purification processes and the activity of the products of each purification step was determined by the avena curvature test. He started this experiment with 33 gallons of the human urine, and final yield was of 40 milligram of crystals. The final product of this process named as oxinotriolic acid or Auxin A. Another active substance was isolated by same method but from corn germ oil by these scientists, and they named it as oxinolonic acid or Auxin B, and it was the same in structure and activity as the Auxin A. These scientists isolated a compound heteroauxin, which is known today indole 3 acetic acid or IAA. This is the structure of indole acetic acid. There are two main categories of these hormones. Some are known as natural hormones and others are known as the synthetic hormones. Natural growth hormones are those which are naturally produced in the plant parts and known as the phytohormones, just like indole 3 acetic acid, 4-chloroindole 3 acetic acid, phenyl acetic acid, etc. And synthetic growth hormones are those which are manufactured in the laboratory by chemical method, although their physiological effects are the same as of natural hormones. Thank you very much.